Today we are going to discuss about oligosaccharides. Now oligosaccharides are the chain of 3 to 10 monosaccharide units. 3 to 10 monosaccharide units. Now we know that on the left side, that is, if 2 monosaccharide units are there, then particular sugar is known as disaccharides, and if more or equal to 11 units of monosaccharides are then it is known as polysaccharides. So today we are going to discuss about 3 to 10 monosaccharide units chain, which is known as oligosaccharide. Now oligosaccharides mostly occur in glycoproteins. Now glycoprotein means it is carbohydrate part and protein part. This carbohydrate and protein are bonded with each other. This bond is actually of the two type. One is O-glycosidic linkage, and other one is a N-glycosidic linkage. And one more thing, that this carbohydrate part is made up of the oligosaccharide. So essentially glycoproteins are the oligosaccharide which is bonded with the protein. This bond is basically of two types. One is O-glycosidic linkage and other one is N-glycosidic linkage. Now if we consider O-glycosidic linkage glycoproteins, then we know that there is a protein chain, right? Suppose this is the protein chain. Protein is basically a amino acid chain, polymer of amino acid. And if in this amino acid chain, if somewhere serine or threonine occurs, then the serine and threonine are OH containing amino acids. So its oxygen atom takes part in the bond formation with the oligosaccharide. Then this particular bond between this protein part and this oligosaccharide part is bonded with the O-glycosidic linkage. Now for the N-glycosidic linkage, there is a protein chain. There is a protein which is made up of amino acid units, polymer of amino acid, and if somewhere in this protein chain, if somewhere aspargine occurs, then this aspargine contain extra nitrogen atom. This extra nitrogen atom takes part in the bond formation with oligosaccharide. Then this particular bond is known as N-glycosidic linkage, and we can call that this glycoprotein is a N-linked glycoprotein. Now this N-linked glycoprotein has a special interest in biology. The reason is whenever this N-linked oligosaccharide is there, N-linked glycoprotein is there, this oligosaccharide shows special properties. This property, the most striking property is it has common pentasaccharide core. This common pentasaccharide core is, there are 3 mannose molecule. This mannose is shown by this circle diagram. These mannose are bonded like this. So these are 3 mannose molecule which are bonded with each other, and 2 N-acetyl glucosamine. So 1 and 2, this triangle shows N-acetyl glucosamine. So whenever we have N-linked glycoprotein, at that time we have common pentasaccharide core. Now this pentasaccharide core may be further extended, like there may be more sugar like fructose and galactose and N-acetyl galactosamine, like that. So here also many sugars are attached, but this pentasaccharide core remains common. Okay, more sugar may be attached with this pentasaccharide core, but this pentasaccharide core remains common. So that means that because of this pentasaccharide core and very different sugar may be attached with this core, it gives us a great diversity of the structure, which further suggests that these information are not only for the structural. Sugar molecule may be giving some information, may be acting as an information macromolecules, just like DNA. So the diversity, this diversity and complexity of oligosaccharide units in this glycoprotein suggests that they are rich in information. So we know that DNA is rich in information. Similar to that, this oligosaccharide part of glycoprotein may also be rich in information. Now, what information this oligosaccharide part provides? So oligosaccharide acts as an Aadhaar card of the cell. We know that in India, in our country, Aadhaar is the identity card of the particular person. So similarly, oligosaccharide acts as an Aadhaar card of the cell. How? Because in human body, there are many cells. So one cell recognizes other cell. So cell-cell recognition takes place because of this glycoprotein, and particularly because of this glyco part, which is in our case, it is oligosaccharide. So for example, if we have hepatocyte, this is hepatocyte, and suppose this is RBC, red blood cell. So hepatocyte recognizes RBC and RBC recognizes hepatocyte. Why? Because on the hepatocyte there are particular types of oligosaccharide present on its cell membrane, and RBC there is different types of oligosaccharide present on the cell membrane. So hepatocyte recognizes this, and further it suggests that this is RBC, and RBC recognizes this oligosaccharide part and recognize particular cell as hepatocyte. So oligosaccharide are very important in cell-cell recognition. So because of this recognition, it is known as Aadhaar card of the cell. Other second most important function of oligosaccharide is it itself acts as a death clock of the glycoprotein. How it acts as a death clock of the glycoprotein? Let's see. If, for example, let's say this is the protein part of the glycoprotein, and this is its oligosaccharide chain. So basically, this wall molecule is glycoprotein. Glycoprotein. This yellow part is of the protein, and this red part is oligosaccharide units. So this complete molecule is of the glycoprotein. Now the peculiarity of this glycoprotein is that it has terminal sialic acid. It has terminal sialic acid, and just beneath the sialic acid there is galactose. Now when any glycoprotein is newly synthesized, let's say it may be antibody or it may be a peptide hormone. These are newly synthesized. For example, antibodies are synthesized in the plasma cell, and this is just now synthesized protein. Then it has this complete structure, which is terminal sialic acid, and just beneath the sialic acid there is galactose. Now once the time passage occurs, with the passage of time, this glycoprotein becomes old, aged, and at some point in the future time, this required destruction, because this protein after some time becomes damaged. Okay, so after certain amount of time, this newly synthesized molecules needs to be destructed. At that time, what happens is this terminal sialic acid. Let me first draw the protein part and its oligosaccharide chain. So this is terminal sialic acid and it's beneath galactose. So this is sialic acid and this is galactose. So the bond between sialic acid and galactose is broken by the enzyme sialase. This bond is broken between sialic acid and galactose by the sialase. So what happens? This galactose becomes exposed on the surface of this glycoprotein. So it looks like this. This is the protein part, and now this is terminal galactose. this galactose is exposed because the sialase enzyme had removed terminal sialic acid. Now, because this galactose is exposed, and on the hepatocyte, if this is hepatocyte, this is liver cell, hepatocyte. On the surface of hepatocyte, there is one special type of receptor which is known as asialoglycoprotein receptor. Asialoglycoprotein receptor, which are present on the hepatocyte. This special receptor recognizes this terminal galactose of the glycoprotein. So what happens? This galactose, which is now exposed on the old senescent glycoprotein, it bonds with this asialoglycoprotein, and its protein part is shown like this. Now, once this glycoprotein is bound with this asialoglycoprotein receptor on the hepatocyte, there is an endocytosis. Endocytosis occurs by hepatocyte. Once this endocytosis occurs, it binds with the lysosome, and the destruction of particular glycoprotein occurs. So essentially we can say that when this glycoprotein was synthesized, it has its biological clock, it has its death clock, which is bound by the sialic acid. Once the function of this glycoprotein is over, the sialic acid was removed, and then once sialic acid is removed, immediate destruction of this glycoprotein occurs. So oligosaccharide part over here acts as a death clock of the glycoprotein. So this is the brief overview about the glycoproteins and oligosaccharide. We will see in the details about the glycoproteins in a separate section. So if you have any doubts, query, confusion, please write it down in the comment section below. Please like, share and subscribe. Thanks for watching.